So today we'll be talking about carbohydrate metabolism. Just a couple of announcements quickly before we get started. Draft day results are posted to CaMKII. The ketone papers will be posted by the end of today for those of you that are presenting on this topic and anyone who wants to kind of get ahead. Beyond that, lecture capture is coming. We'll be on top of it now, just working through some technological and software related difficulties. So today's lecture will be talking through an introduction to carbohydrates before moving on to glucose uptake. Then we'll move into talking about the regulation of glycogen metabolism, whether that is glycogen synthesis or glycogen breakdown, before finishing off with glycolysis, so glucose metabolism, and the different pathways that we can go through for this, whether that is anaerobic or aerobic metabolism. So again, just a quick overview. What we can see here is this figure we're going to see time and time again. And really the idea I want to focus on today is glucose metabolism, whether that is bringing glucose into the cell, so into the cytosol, or whether we're talking about glycogen breakdown that occurs within the cytosol to promote ATP formation through pyruvate. So quickly moving into some of our carbohydrate stores. So thinking about the major store we have in the body, that is muscle glycogen. So about 400 grams of glycogen is stored here, and that's 80% of our total glycogen storage. Beyond that, we have 100 grams stored in the liver, and a smaller amount stored as blood glucose, only about 3 grams. For a little bit of perspective, we know that we have about 503 grams of glucose or glycogen within the body, accounting for about 2,012 calories, since we know there's 4 kilocalories per gram of carbohydrate. And I want to highlight that these stores are finite and expendable because we can actually expend 2,000 kilocalories with about 1.6 hours of intense exercise, or if we ran 32 kilometers. So I just want to highlight again that these stores, we're able to run through them, which is why it's so important that we're able to resynthesize, rebuild in a post-exercise period. Next, I want to give some perspective. So 128 grams of sugar are in a large DQ blizzard. So you have about 3 large DQ blizzards within your whole body stored as glycogen. So again, not that large of a store. Moreover, carbohydrates can be metabolized both aerobically and anaerobically, and we'll get into that in the latter half of this lecture. First off, I want to talk about some important lingo with regards to carbohydrate metabolism. First and foremost, we'll talk about things related to genesis. When we're talking about genesis, we're talking about synthesizing or building, whether that is gluconeogenesis, or synthesizing glucose, or glycogenesis, where we're synthesizing glycogen. Moreover, when we talk about lysis, we're talking about breaking something down. So if we talk about glycolysis, we're breaking down glucose, and we'll get into this later in the lecture, but we're really focusing on that committed step to glycolysis, so the formation of glucose 6-phosphate from glucose. And when we talk about glycogenolysis, we're talking about the breakdown of glycogen to provide individual molecules of glucose. Importantly, none of these terms are interchangeable, meaning that they do not mean one another. We can't use them in different contexts. If we use them in the wrong setting, it makes the statement obviously wrong. So first I want to talk about glucose and glycogen. So glucose is a 6-carbon molecule, can be used as an energy source, and we know it's important as a fuel for exercise, whether that is the onset of exercise or during high intensity exercise bouts. Alternatively, glycogen is basically the storage form of glucose, so it's a polysaccharide, meaning it's comprised of 3 or more sugars, and again, it represents that storage form of glucose, where we actually have individual glucose molecules attached to one another via glycosidic bonds. So glycogen is in this branched shape, and the reason that we get this is through, 1, glycogenesis, so that building of glycogen is catalyzed by the enzyme glycogen synthase and something called glycogen branching enzyme. We're going to learn that basically this branching shape is beneficial for glycogen's use and storage. But really, glycogen can range from a few hundred to up to 30,000 glucose molecules, so it can be quite small or it can be absolutely massive. Glucose can be added or taken away one glucose molecule at a time from glycogen. And that's occurring where the arrows are shown here, so at the terminal ends. So what's the benefit of glycogen branching out or being in this large branch shape? First and foremost, when we consider glycogenesis or glycogen synthesis, we're building that glycogen, it actually increases the rate of glycogen synthesis, meaning we can add those glucose molecules wherever on those terminal ends, and it's not really picky. Second, when we're talking about glycogenolysis, we're talking about glycogen breakdown and the idea that we can increase the rate of breakdown if we're taking it away from anywhere on those terminal ends. Lastly, glycogen can have increased solubility due to this branching structure because we're able to basically create this hydrophilic surface that promotes its ability to dissolve in fluid. So what are the fates of glucose? Where does it go once it's within the body? If we're in a fasting state, a majority is taken up by the brain and used to fuel cognitive processes. And 25 to 45% is used by the kidneys, the gut, red blood cells to maintain proper physiological function. And the last 15 to 25% is used up by skeletal muscle. The key point of this slide is that in response to large carbohydrate loads, so a carbohydrate-rich meal, for example, or a glucose tolerance test, 70 to 80% of this glucose is taken up by skeletal muscle. So skeletal muscle is a major sink for glucose when it needs to be taken up and stored. I said I wasn't going to talk a lot about the liver, but we'll take some time to go over glucose metabolism in the liver as it relates to its storage as well as its release. It's important to consider and acknowledge that this storage site exists, and we can kind of relate that to how it's important for the skeletal muscle as well. So the liver serves as a storage reservoir for glucose. In that regard, it can store glycogen via glycogen synthase, and it can rapidly provide glucose when it's needed. So if we're in a case of starvation or at the onset of exercise when we need more glucose in the blood, we can actually provide it from the liver. And there's two ways that this can occur. One is glycogenolysis, so breakdown of stored glycogen in the liver. This dominates early in exercise when we need to maintain those circulating blood glucose concentrations, or we can have gluconeogenesis, so the formation of glucose from non-glucose precursors. These non-glucose precursors are alanine, glycerol, pyruvate, and lactate. And this dominates later in exercise because we need time for those precursors to be formed. So alanine is an amino acid, glycerol comes from the breakdown of triacylglycerol, pyruvate we'll see is a product of glycolysis, and lactate is going to be a byproduct of exercise metabolism. So again, the key point here is that gluconeogenesis dominates later in exercise, as well as glycolysis dominating early in exercise, being the two forms of glucose release from the liver. And the reason we have this METABOLISM symbol in the top corner is really the idea that the liver provides glucose to maintain blood glucose concentrations in response to a threat in a drop in those blood glucose concentrations. So examples, again, fasting, starvation, or exercise. Here I want to look at glucose uptake and storage in the liver. So first and foremost, a couple things I want to highlight here. You guys don't need to memorize the steps involved in glucose uptake and storage in the liver. I just want to highlight a couple key differences that exist between skeletal muscle, as well as a similarity in the fact that, again, the end goal for glucose storage as glycogen is that we're going to be adding a glucose molecule to glycogen. Same thing happens with glycogen storage in skeletal muscle. Key differences, though, we'll see that GLUT1 and GLUT2 are the transporters involved in glucose uptake as well as glucose released back into the circulation through GLUT2 for the liver. And importantly, glucose 6-phosphatase is present in the liver but not in skeletal muscle. The purpose of glucose 6-phosphatase is to take glucose 6-phosphate and turn it back into glucose. By doing this, we're able to shuttle glucose from the liver back into the circulation. This does not exist in skeletal muscle. Once glucose is turned into glucose 6-phosphate in skeletal muscle, it is stuck within the skeletal muscle, or it is trapped within that skeletal muscle. So now here's our first example of talking about the regulation of glycogen synthesis and breakdown. For this, we're talking about the regulation of liver glycogen synthesis. And really what we need to do when we're talking about the regulation of human metabolism is thinking about, in a logical manner, why the certain molecules or the certain stimulators or inhibitors are doing what they're doing. So first and foremost, glycogen synthase, its role is to synthesize glycogen. So we want to build up glycogen with glycogen synthase. Glycogen synthase can exist in a more active or a less active form. The more active form is caused by dephosphorylation, and the less active form is caused by phosphorylation. So what we can do here is look at the different regulators of glycogen synthase and kind of make sense of how they regulate or they change glycogen synthase activity. So first and foremost, if we look at insulin, we're going to learn that insulin is anabolic. So insulin wants to build. So insulin will cause an increase in glycogen synthesis, or an increase in glycogen synthase activity. With that being said, we can see that insulin inhibits the conversion of glycogen synthase into the less active form, and insulin also stimulates or promotes the conversion of glycogen synthase into the more active form. Alternatively, when we think about epinephrine and glucagon, so epinephrine is that stress response typically seen at the onset of exercise, and glucagon is a signal to raise blood sugar. If we want to raise blood sugar, we're going to need more glucose. If we want fuel for exercise, we need glucose to be broken down into ATP. So here we're going to see less synthesis activity or less activity of glycogen synthase. And how that's promoted is through epinephrine and glucagon stimulating cyclic AMP. Cyclic AMP then goes through its own signaling cascade to stimulate the conversion of glycogen synthase into its less active form. Alternatively, epinephrine and glucagon can also cause a decrease or an inhibition of the conversion of glycogen synthase into the more active form. Again, epinephrine and glucagon are signals for the breakdown of glycogen into glucose. So in these cases, we'd be converting glycogen synthase into its less active form. Here, I just want to highlight a couple points. So we're talking about liver gluconeogenesis. So we mentioned that late in exercise, the liver can actually create glucose. And what's happening here is I just want to highlight the point that our precursors, lactate, glycerol, and amino acids, are going to be formed as products of exercise metabolism that can then be used late in the exercise period to produce glucose through liver gluconeogenesis. Importantly, the role of cyclic AMP here. So we saw in the last slide that cyclic AMP promoted glycogenolysis. So same idea, we want to break down or form glucose in order for ATP production. If we want that, we probably would equally want the synthesis of glucose through gluconeogenesis. So now moving into skeletal muscle, talking about glucose metabolism, specifically storage and utilization. The first thing we need to talk about are the specific glycogen pools in skeletal muscle. So it's a little bit of an oversimplification to say simply that glycogen is stored in skeletal muscle. This is true, but we can actually have specific regions with which glycogen is stored. So this is a bit of a throwback to muscle physiology. First, we need to talk about the fact that a skeletal muscle is comprised of fascicles, and fascicles are basically bundles of muscle fibers. Then we need to think about the fact that we have single fibers, which are encased by something called the sarcolemma, and this is within that fascicle. Within each single fiber, we actually have thousands of myofibrils. So that's kind of the level of breakdown that we're looking at for the skeletal muscle. And what we'll see is that there's different glycogen pools within the single fibers. So within each single fiber, we have three different pools. One, subsarcolemmal. So remember that sarcolemma is wrapping around the single fiber, and we can have glycogen stored directly beneath that sarcolemma. Additionally, remember each single fiber is made up of thousands of myofibrils. We can have glycogen stored between the myofibrils. We can also have glycogen stored within the myofibrils. So the three major stores again are subsarcolemmal, so beneath that sarcolemma, between the myofibrils, so intermyofibrillar, or intramyofibrillar, which means that they're actually stored directly within each myofibril. While we're on the topic of muscle fibers, another thing to touch base on is the idea of muscle fiber types. So research out there exists where they look at electrical stimulated muscle contractions. And what they found is that, one, at baseline, glycolytic muscle fibers, so in this case, in a rodent, that would be the extensor digitorum longus that's typically used. Baseline glycogen content is higher in this glycolytic muscle compared to the oxidative muscle. So what we can see is we have to look at the y-axis on these graphs and see that muscle glycogen goes up to about 7 nanomoles per milligram in glycolytic muscle, whereas it's only about 4 nanomoles per milligram in oxidative muscle. So the stores of muscle glycogen are higher in glycolytic muscle to start. And following skeletal muscle contraction, or this period of skeletal muscle contraction, we have a greater rate of glycogen utilization in glycolytic muscle. Specifically, we can see that in the extensor digitorum longus, our glycolytic muscle, muscle glycogen concentrations drop to about 2.5 nanomoles per milligram compared to about 3 nanomoles per milligram in the oxidative muscle. Thinking about glucose uptake in skeletal muscle. So skeletal muscle, again, is this major sink for glucose because skeletal muscle comprises about 40 to 60% of a mammalian's body mass. As opposed to the liver, the major glucose transporters in skeletal muscle are GLUT1 and GLUT4. Specifically, we'll talk a lot about GLUT4. And the main stimulators of muscle glucose uptake are going to be insulin-mediated as well as contraction-mediated pathways. So this is going to be important because in order to use glucose for fuel, or in order to store muscle glycogen, we have to actually be able to get it into the skeletal muscle cell. What I want to highlight here is that now that we know there's insulin-stimulated and contraction-mediated glucose uptake, I want to highlight the fact that these are additive pathways. So we see that insulin stimulates skeletal muscle glucose uptake. Contraction also stimulates skeletal muscle glucose uptake. And together, there's this additive effect where there's even greater skeletal muscle glucose uptake with both insulin and contraction-mediated pathways together. What this suggests is that they're actually occurring through separate mechanisms. And because of that, we can actually delve into the different mechanisms that exist for how insulin and contraction-mediated pathways work. So we'll start with glucose uptake through insulin-stimulated pathways. First and foremost, insulin is Canada's gift to the world. It was discovered by Banting and Best in 1921, actually out of the University of Toronto, and they won the Nobel Prize in 1923 for this invention. Insulin is a polypeptide hormone secreted by beta cells of the pancreas. What it actually does, it stimulates glucose uptake in insulin-sensitive tissues. So we're going to be focusing on skeletal muscle as well as adipose. And what happens is that glucose uptake occurs through the translocation or the movement of glucose transporter, so GLUT4, from intracellular sites to the plasma membrane. So when would we see increased insulin levels? So in healthy individuals, blood glucose should rise after a meal. It should rise from about 5 millimoles to 9 millimoles following a meal. In diabetics, it can actually rise up to 20 millimoles, and we'll kind of discuss the pathology of diabetes in a few slides. So insulin is an anabolic hormone, and what we're really focusing on in today's lecture is the idea that it promotes glucose uptake and glycogen storage. In the coming weeks, we'll see how insulin can promote fat synthesis and storage as well, and protein synthesis too. So big picture short, or the long story short, is really the idea that insulin-stimulated glucose uptake is mediated through GLUT4, and in order to get GLUT4 at the plasma membrane, we need a phosphorylation cascade to occur. So insulin binds to its insulin receptor and causes this phosphorylation cascade that brings GLUT4 from its intracellular sites to the plasma membrane. Glucose is uptaken, and once its job is done, that glucose transporter, that GLUT4, is brought back into its intracellular sites until it's needed again. So how does this actually occur? So that insulin-stimulated glucose uptake is mediated through GLUT4 via a phosphorylation cascade that really in the end phosphorylates AS160, which can then move GLUT4 to the plasma membrane. But really, this is oversimplified. There's a lot more going on between insulin binding to its receptor and AS160 activation, leading to GLUT4 translocation. So if we take a closer look at the insulin receptor itself, what we see is that it spans the cell membrane, and it consists of 4 subunits, 2 alpha and 2 beta. The alpha subunits protrude into the extracellular space, communicating with the environment around it, and the beta subunits protrude into the intracellular environment, closer to the skeletal muscle GLUT4 pools. So once insulin binds to the alpha subunits, what actually happens is that the beta subunits become active, and they can actually autophosphorylate themselves and become active. Once they become active, insulin receptor substrate 1, or IRS1, can actually dock onto the beta subunits. Following this docking, what happens then is that IRS1 can serve as a docking site for phosphatidylinositol 3 kinase, otherwise known as PI3 kinase. The activation of PI3 kinase then causes the subsequent phosphorylation of phosphatidylinositol bisphosphate. There's an asterisk here because I had a typo on the lecture slides that you guys have, and which is also known as PIP2. PIP2 can then be used to create, through its phosphorylation, can be used to create phosphatidylinositol triphosphate, so PIP3. Importantly, what happens then is that PIP3 can then activate one of 2 pathways. It can either activate protein kinase B, which is also known as AKT, or it can activate protein kinase C. Ultimately, this leads to the phosphorylation of AS160. AS160 we know is responsible for bringing GLUT4 from those intracellular pools to the plasma membrane. Now we see that there's an asterisk on PKB or AKT here. And what's kind of important about that asterisk is that AKT doesn't just directly act on AS160. It's a little bit more complicated than that. So if we delve into it a little bit further, what we see is that PIP3 recruits AKT to the plasma membrane. Here it can be phosphorylated by serine 473, or phosphorylated at serine 473 by TORC2 kinase. This causes a conformational change in AKT, which allows for another kinase to phosphorylate AKT at threonine 308. Now that AKT has been duly phosphorylated, it can come back to AS160, phosphorylate AS160, allow that translocation of GLUT4 to the plasma membrane. So now I want to talk about insulin signaling in the context of diabetes or metabolic disease. So what we see is that in diabetic individuals, we actually have the same total GLUT4 content compared to healthy individuals. But what we see here is I actually want to compare lean animals versus the ZDF or Zucker diabetic fatty animals. And what you see is that C is for control, H is for high fat, where it says MET is metformin, an anti-diabetic drug, and then X is for exercise. Strictly looking at the control, high fat, and high fat with metformin groups compared to the lean animals, what we can see is that there's no change in GLUT4 content in lean versus diabetic rats. What we see, the change is really based off of the provision of exercise, a known stimulator to increase GLUT4 content. So again, there's no difference in total GLUT4 content between healthy and type 2 diabetic rats. What we really see is that there's a difference in the way that lean versus type 2 diabetic rats can activate the insulin signaling cascade. So basically, GLUT4 translocation is impaired in these animals. So their ability to respond to insulin is impaired, although their GLUT4 content remains the same. Importantly, in these individuals, contraction-mediated glucose uptake through GLUT4 translocation is preserved, and that's half of the premise for case study number 1. So we're thinking about the carbohydrate side of this equation and how contraction-mediated glucose uptake is actually preserved in type 2 diabetics or anyone with metabolic syndrome. So how do we actually test insulin-stimulated glucose uptake? One of the main ways we do this is through a glucose tolerance test, where we can provide a bolus of glucose and track blood glucose over time. So in humans, how we do this is through the ingestion of a sugar beverage called Trutol. It's about 75 grams of sugar, and it tastes like flat orange crush. In mice, we can either deliver an oral glucose gavage, meaning that we deliver it directly through the mouth, or we can deliver an intraperitoneal injection of glucose, which is 2 grams per kg body weight. One thing that we talked about in class is this idea of a difference in the response with a gavage versus intraperitoneal injection. And these are the things we should be thinking about when we're talking about our presentation papers and methodologies used for research. The difference with gavage versus intraperitoneal injection is that oral delivery of glucose actually stimulates an intestinal response with the release of incretins, which can actually stimulate insulin secretion, meaning that if we would have a larger insulin response with an oral glucose gavage versus an intraperitoneal injection. Again, these are just some things to think about when we're talking about study design and research methods. So when we looked at 20 weeks of a high-fat diet where 60% of the kcals come from fat, we see in these rats that they have an impaired glucose tolerance. And we know that because in the white dots with our high-fat diet compared to the black dots with our low-fat diet, their ability to clear the glucose following that glucose challenge is impaired. Again, that area under the curve is significantly higher in these high-fat fed animals. And the only problem with the glucose tolerance test is really the idea that we don't know how much insulin was used to clear this glucose load. So another thing that we can do in conjunction with a glucose tolerance test is called an insulin tolerance test. In an insulin tolerance test, we're again delivering a standard bolus of insulin in this case, and it's actually delivered intravenously or intraperitoneally, and blood glucose levels are monitored over time. The goal of an insulin tolerance test is to induce hypoglycemia. So we want to give insulin to promote glucose uptake, and usually this is done in a fasted state so that we achieve those hypoglycemic levels. As discussed in class, we can't deliver insulin via gavage or orally to humans because insulin is actually broken down and digested, so it has to be delivered intravenous or intraperitoneal. Again, we see the same case of that 20 weeks of a high-fat diet, 60% of the K comes from fat. We see impaired insulin tolerance in these high-fat fed animals. We know that because following that delivery of glucose, they have a higher area under the curve or an inability to take that glucose up into the skeletal muscle tissues, meaning that they're less responsive to insulin and they can be termed insulin resistant. Now I want to talk about contraction-mediated glucose uptake. So we know the outcomes of skeletal muscle contraction. 1, ATP is hydrolyzed to allow for cross-bridge cycling, meaning that ATP is broken down into ADP and further into AMP. Increasing these levels of ADP, AMP can basically stimulate something called AMP-activated protein kinase, otherwise known as AMPK. AMPK is a cellular energy sensor, meaning that when ATP levels are low or ADP and AMP levels are high, it becomes activated to stimulate carbohydrate and fat uptake as well as its oxidation. So we see in this pathway here, exercise increases free ADP, free AMP. Again, the cell senses, AMPK senses these high levels of breakdown products of ATP, activates AS160, brings GLUT4 from its intracellular stores to the plasma membrane to promote glucose uptake to be used to produce ATP eventually. So what else happens during contraction? So during skeletal muscle contraction, we know we see a rise in calcium levels because calcium binds to troponin. It relieves that tropomyosin blockade of myosin sites on the actin filament, and cross-bridge cycling occurs. We know that calcium can also bind to calmodulin. So calmodulin is this molecule up in the top corner here, where calmodulin can bind 4 calcium ions. When calcium binds to calmodulin, it becomes activated, forms this calcium-calmodulin complex. And basically what happens then is it can activate calcium-calmodulin-dependent kinases, or CAMKIs. Examples of these are calcineurin or myosin light chain kinase. CAMKII specifically can have a role in the regulation of glucose metabolism. On the acute scale, previous research has shown that CAMKII reduces contraction-mediated glucose uptake, but does not affect insulin-stimulated glucose uptake. On the chronic kind of scale, chronic activation of CAMKII can enhance glucose transport, and this is mediated through increased GLUT4 protein content. The way that acute and chronic activation of CAMKII is mediated works through something called the memory property of CAMKII. So how that memory property actually works is that contractile stimulation, so skeletal muscle contraction, causes these increases in calcium levels. Calcium activates calmodulin, activates CAMKII. CAMKII, once it's activated, has its own autophosphorylation, so it phosphorylates itself. And basically, with repeated muscle contractions, we can have these effects that persist over time because the phosphorylation hasn't worn off from the previous contraction yet, kind of like graded action potentials or action potential summation. So again, repeated contractions can cause this effect to last over time because that phosphorylation kind of builds up and persists over the time. So why would repetitive increases in cytosolic calcium increase glucose uptake? So what we're thinking about here with skeletal muscle contraction is the idea that calcium is released and at the end of the contraction has to be reuptaken into the sarcoplasmic reticulum by sarcoendoplasmic reticulum calcium ATPase, otherwise known as SERCA. Because SERCA is working against this concentration gradient, this requires ATP. So with each contraction, we're actually breaking down ATP, resulting in increased ADP and AMP. So as we mentioned in class, increased ADP, increased AMP, or decreased ATP can actually signal for AMPK to come to the rescue to basically increase GLUT4 translocation and bringing more glucose into that cell.